Now, when we have finished studying the body fluids and blood particularly, we start to consider the principles of regulation of the body fluids. And first to be considered is the so-called humoral type of regulation that is achieved through release of chemical signals into body fluids, mostly blood. Greek word humor exactly means fluid, so this humoral type of regulation developed early in evolution. So let's begin to consider principles of information exchange between cell and the environment. Simplest unicellular organisms constantly maintain this exchange, and the cells of a multicellular organism also maintain the exchange with the environment with the following points: substance, energy, and information. This exchange is required for integration of the cell functioning and also is necessary for the survival of the organism. And information that can be transferred is transferred by means of various signals. Which type of signals exist? We divide them on the principle of which nature the signal is. Signals of physical nature. They include such examples as light, sound, pressure, temperature. Signals of chemical nature. This is a very large group of chemical signals such as hormones, neurotransmitters, cytokines, growth factors, odorant molecules, flavor molecules, and so on. Then there is a group of combined physical and chemical nature signals. We studied this already, and these are osmotic pressure, oncotic pressure, hydrogen index, pH, electrical potentials we are going to study in future, partial pressure of gases, and so on. And there are complex signals, such as combination of different sounds, different odors, colors, and so on. And there is a specific complex signal, specific for humans, word, special kind of signal that is highly involved in the process of information transfer. To be a true signal, the signal should be perceived. Let's say we have a signal. To detect the signal, we need to have the certain detecting device, some receiver, to get this signal. And our receptors exactly are such detecting devices. Receptor is a structure that perceives signals. The name receptor comes from Latin recipe. This word you're going to use many times when writing prescriptions. In abbreviation RP, you will say to take some tablets, some ampoules, some other drugs, and so on. This is a widespread used word that came from Latin language. Receptors that take our signals and transfer them. And for receptors, we have two big basic types, two classes of receptors, which are very different. First type is molecular or cellular receptors. These are complex molecules that are built in the cellular membrane or somewhere intracellularly, and these receptors serve for perception of various chemical signals by the cell. So these receptors are intended for control of the cell functioning, for regulation of the particular cell in which receptors are located. And there is another type, a different type, other type, sensory receptors that are specialized complex cells that are designed for perception of the sensory signals of various nature. And then their task is to transform these signals into nerve impulses, which can be sent further to the central nervous system. And this is a general, universal way to inform the nervous system by the sending of nervous impulses or action potentials. And then it provides the sensory input to brain for further integration and analysis of information. So today we are going to concentrate on molecular receptors that enables us to control our cells' functioning. It's not the only one way, but it's an important part of regulation, hormonal regulation. In contrast to nervous regulation, which also is going to be studied later. Receptors are involved into most types of the intracellular communication, most types. Basic types of intercellular communication between cells are the following: auto and pericrinia, then endocrinia and subtype neuroendocrinia, synapses, and gap junctions. All these types we are going to consider in detail. First, the Greek root krinia means release, release somewhere. For endocrinia, this is release into internal environment of the body. Let's see. First three types involve molecular receptors, and just gap junctions do not need receptors. Now let's consider which types of intercellular communication the receptors take part in, which type. First, let's see, this is a cell with nucleus, and somewhere in the membrane of the cell we see molecular receptor, schematic presentation. And signal can be shown like this circle, and let's imagine that cell produces this signal molecule, like hormone or neurotransmitter or something else, some biologically active substance. And the cell has molecular receptors that is able to bind exactly this signal molecule. When cell releases this molecule, it can be bound to this receptor. So it's a case when cell influences itself, auto, directed to itself. So the cell releases something, krinia, and makes effect on itself, autocrinia. The same practically, just little bit different type is the pericrinia, when cell releases substance and it binds not to the same receptor, the same cell receptors, but to receptors of similar adjacent nearby located cell, which possesses similar receptors correspondingly. So this case is pericrinia. The peri means nearby, about, around. So pericrinia and autocrinia are just subtypes of one similar type of communication, which acts on a short distance, only to nearby located cells, somewhere in an interstitial space perhaps. Then let's consider long distance communication. This is endocrinia, which is performed by endocrine cells. These cells are specialized to release substances into blood. And with bloodstream, molecules can be transported into any distances within the body and reach any distant cell. Just this target cell, if it's target cell, it should possess receptors exactly for the substance released from endocrine cell. Target cell with receptor. Look, the signal molecule is released, and when it's released with bloodstream, it reaches the receptors of the target cell. In this case, we can say that this signal molecule is a hormone, because it's classical criteria for hormone to be secreted into blood. So now the hormone influences distant cells easily, reaching it with blood. This is endocrinia, and the cell that releases hormone is exactly cell and with endocrine function. But there is also subtype when this is not simply endocrine but specific neuroendocrine cell, neuron that has the endocrine function. And in this case, the signal molecule is released from the end of axon of this neuron. And target cell also can be located distantly, because release occurs again into internal environment. So substance is released from the axons, produced somewhere in the body of the cell, is transported along the axon, then reaches the end of axon and released into blood. With blood, it reaches finally the target cell, binds the receptor, and influences the cell in a specific way. This is subtype of endocrine, neuroendocrine. Difference is just the different original cell that releases this signal molecule, simply endocrine cell or specific neuroendocrine cell. In all these considered cases, there is a molecular receptor necessary for perception of the signal. So receptor is necessary. Then next type of communication between cells, the synapse, which belongs generally belongs to the electrical signaling, which developed later. But even in the electrical signaling, we use the chemical substance that is released from the synaptic terminal. Here the neuron can be seen, that the innervated cell, which can be other neuron, the muscle or endocrine cell. And here release of substance occurs from the end of axon, synaptic terminal, but it's not transported from the body and immediately finally released, but it's concentrated in the terminal of the synapse, synaptic terminal, the end of axon, and release occurs under stimulation by nervous impulse. Nervous impulse generally is produced somewhere in the beginning of an axon, which is axon hillock, the beginning of the initial part of axon. And when nervous impulse is generated, it quickly travels along the axon, reaches the end, and here it stimulates release of a chemical substance. We say this is a neurotransmitter. And this neurotransmitter, after release, easily diffuses very short distance to the next cell with its receptors and binds to this receptor. So we see even electrical signal actually requires some chemical signaling elements involved. This is synapse, and synaptic contact between neurons also has the receptors on the membrane, which is called postsynaptic in this case. And actually it looks like the following. The dilated portion of the synaptic terminal almost touches, but really does not touch because there is a cleft, the membrane of the next cell, and membrane of the next cell contains numerous receptor proteins. Basically, receptors are built of proteins, not only these, but many others. And finally, gap junction. Here it's the only case when we don't need receptors, because gap junctions are the special channels that connect membranes of the two neighboring cells. Simply directly, like a tube inserted through two very tightly connected membranes. And you may remember from biology that these channels are formed by specific connexons, which make subunits, and finally they make direct tubes that inserted into membranes. And through these tubes, through these gap junctions, fluid may easily flow, and this fluid contains electrolyte charged substances. Therefore, charges easily can be transmitted from membrane of one cell to the next one. And so membrane potential can be changed. When one cell changes potential, it immediately changes the potential of nearby located cell very quickly, at least. You know, such gap junctions exist in different organs, and for example, they are present in the smooth muscle cells, and also they are present in the cardiac muscle. They are known under the names nexuses or intercalated discs for the heart. And through these cells get impulses from each other and quickly transfer these impulses. So, and now we are going to concentrate on molecular receptors involved into most types of communication between cells. Receptors, basically, as I said, are molecules of protein origin inserted into membrane. And these receptors are specific and are able to bind very specific types of substances. And these substances, in this case, are called ligands. Regardless of their nature, it can be hormone, neurotransmitter, biological active substance, cytokine, and so on. But they all are able to bind. And if they are able to bind, their general common name is ligands. You know, from the root, from Latin, ligament, this root means something combining, attaching. So ligands are substances that bind to receptors. And let's begin with classification of molecular receptors. This classification divides them into two groups, big groups, and then subdivided. First big group is receptors of the cellular membrane or plasma membrane. Notice plasma membrane is a simply usual cellular membrane. Don't have any mix with blood plasma, which is fluid and not related to membrane. So it's a classical outer membrane of the cell with bilayer of phospholipids. And among these phospholipid molecules, the receptors can be located. These are cellular receptors. They can be of the three types. First, they are called according to their structure. This type of receptors is called one transmembrane segment receptor. So the abbreviation for transmembrane segment is TMS. Therefore, it's 1-TMS. It's clear the molecule, protein molecule, passes through the membrane just one time. So the only one segment. Simpler constructed receptors. Then much more complex are 7 transmembrane segment receptors. Of course, their main difference is not only the number of segments, which is high, but they operate and function differently, and we are going to consider these differences. So 7-TMS receptors are very much widespread. And the third type is ligand-gated ion channels. Ligand-gated means that ligand, after binding, opens gates of these channels, and channels become open. No ligand channel is closed. And second type of receptors is located inside the cell, intracellular receptors. They are divided into nuclear and cytosolic. But here difference is not as great as difference between types of cellular membrane receptors, because it doesn't matter, nuclear or cytosolic receptors they anyway operate through processes in the nucleus based on DNA transcription and formation of proteins. So even cytosolic receptors later enter the nucleus and then have common final pathway of operation, nuclear and cytosolic receptors. Ligands. Before consideration of each type of receptors, we should understand why some ligands bind receptors inside the cell and some ligands only bind from outside. Any molecule that is able to bind to the receptors is a ligand, and molecular receptors recognize and bind specific ligands. So one type of receptor only for corresponding type of ligand. And binding results in signal transmission. Ligands include hormones, neurotransmitters, local chemical factors such as growth factors, cytokines, and others. And first we divide the ligand by their nature, hydrophilic or lipophilic. Hydrophilic are dissolved easily in water because they are charged. They have charged portions. So between H plus and OH minus, they can be dissolved. Lipophilic substances do not have charges, and therefore they are insoluble in water, but oppositely they easily become dissolved in lipids. And if you remember that membrane consists of phospholipids, then it's easy to understand that hydrophilic ligands are not able to pass through the membrane, while lipophilic can do it. And that's why we have this division of molecular receptors into two big groups. Lipophilic substances do not need outer surface receptors. They simply enter the cell through membrane, being dissolved among phospholipids, and they bind to intracellular receptors. And hydrophilic substances are not able to pass through membrane, so they need to have their receptors designed for them located on the outer surface of the cell. And exactly hydrophilic substances bind to these receptors, but through these receptors signal is transmitted through the membrane and reaches the interior of the cell and changes the functioning of the cell. Actually both types of ligands, lipo and hydrophilic, they bind to different types of receptors, but their task is finally to change something inside the cell. Just intracellular receptors provide for lipophilic ligands to do everything themselves, but hydrophilic substances should have some helper inside the cell. And among these helpers, we can find some substances that are called second messengers, not for any type of hormones we'll consider. Receptors that are lipophilic include steroid hormones, thyroid hormones, and vitamin D3. Subtype D3 active vitamin is lipophilic and can be considered as hormone. Therefore, we include here to the group of steroid and thyroid hormones. All steroid hormones are based on cholesterol and they are easily dissolved in lipids. Thyroid hormones, regardless of their derivation from the tyrosine amino acid, nevertheless have such a structure that they are lipophilic. And most of hormones of protein and peptide nature belong to hydrophilic. Proteins generally are hydrophilic. And so proteins, peptides, they all use the receptors of plasma membrane of the cell, or simply cellular membrane receptors. Let's begin to consider in details receptors with one transmembrane segment. Their basic structure is shown here. For example, this is EGF, epidermal growth factor. Anyway, these receptors have the outer segment, which is extracellular ligand binding domain. Then they have their single transmembrane segment that passes through the membrane, and inside, it's very important that they are connected to the intracellular tyrosine kinase domain. And so here we see kinase. This is an enzyme portion of the molecule that is able to produce corresponding biochemical reactions. And it's inactive without ligand, and when ligand binds to the receptor from outside to the extracellular ligand binding domain, intracellular kinase starts to work. So this is a change that we expect for the cell. Another example is insulin receptor. Do not look at as it looks from first attempt that it's like two receptors, two segments receptor, but really this is combination of the two receptors, and for insulin it's the usual way of activation. Two receptors should be joined. Basically, structure of each receptor for insulin is similar. Again, there is extracellular ligand binding domain, one segment that passes through membrane, and intracellular kinase. Just for the insulin receptor, it's necessary to join, dimerize two receptors together. And then tyrosine kinase becomes activated. When it all occurs when insulin binds to the receptor from outside. And I need to say from the beginning that actually insulin is the only one classical hormone that uses this type of receptor. The other ligands for this receptor are mostly the different growth factors, like you see example from the left side. It's like cytokines, the hemopoietic factors, they all operate through One-TMS receptors, but they are not hormone, they are not released into blood, and they mostly operate within short distances. Kinases. We mentioned the tyrosine kinase in the One-TMS receptor. That generally kinases. This is a great group of enzymes which perform one specific reaction. They accomplish phosphorylation, addition of the phosphate group. This phosphate group initially taken from ATP molecule, and it's transferred to the necessary substrate protein molecule, substrate molecule that undergoes transformation. So phosphorylation, addition of phosphate group, is very important way of regulation for all intracellular protein. And if enzyme protein is not active, then phosphorylation makes it activated enzyme. If it's a structural protein, the necessary desired change of its conformation, therefore and function, is achieved through phosphorylation. Generally, it occurs in such a way that in our body that proteins for their activation or specific change require phosphorylation. So the key enzymes for these are kinases. And kinases comes from the movement, so they transfer the phosphate group. The opposite reaction of removing of phosphate group performed by phosphatases, phosphate splitting, phosphate splitting enzymes, phosphatases. It's opposite reaction, so we constantly operate phosphorylation and dephosphorylation, these two group of enzymes. Look, if it's a structural protein which is initially inactive enzyme or structural protein that does not have necessary conformation, when kinase starts to operate, it takes also ATP molecule and combines this protein with one phosphate group. And ATP, adenosine triphosphate, becomes converted to adenosine diphosphate in this case. And so it uses energy, as you can see, for each reaction. But then phosphorylated, with letter P, it's phosphate. Phosphorylated protein becomes activated enzyme or changed structural protein. And so phosphorylation and opposite dephosphorylation, these two reactions are the main way of regulation of cellular protein functioning. Functions of one-TMS receptors, as I already started to mention, mostly include stimulation of processes of cell growth, cell division, differentiation, induced by cytokines and growth factors. And more exact examples are stimulation of nerve growth, vessels growth, antibody synthesis stimulation for immune response, and hemopoiesis regulation, direction of differentiation of stem cell into specific way, into specific cell conversion. Then blood coagulation reactions, control, phagocytosis, direction and stimulation, control of cellular adhesion, and so on. And another example also comes from the insulin function, stimulation of synthesis of carrier protein. For example, transporters for glucose. Glucose transporters exist in different subtypes, and insulin induces formation of one of such subtypes. And glucose transporters are abbreviated as GLUT. So it's also a function of one-TMS receptors. Let's move to the 7 transmembrane segment receptors. These receptors' full name, as you can see, includes also the ending, coupled with G proteins. If you remember about this, you will never make a mistake in comparison of 1-TMS to 7-TMS receptors. It's important part, coupled with G proteins. G proteins is a specific type of proteins that are able to bind GTP. Full name is GTP, guanosine triphosphate binding proteins. But usually no one says such a long way of description, simply G proteins. This term is used. Here you see two examples of the structure of these 7-TMS receptors. These are two types of adrenergic receptors, receptors for adrenaline. One side you see beta adrenergic receptors, the other alpha adrenergic receptors. But both have similar, something in common. They have extracellular ligand binding domain, then they have their 7 transmembrane segments. And by the way, they are not necessarily arranged like in this schematic presentation in line. No, they usually make a bunch of these segments, so it is a compact structure. But here, for simplicity, you can see and even count these exactly 7 segments that are lined inside the membrane. And in the inner side, there is intracellular domain, which is connected to G protein. G protein itself is not shown here. So, of course, their structure is much more complex, but functioning is even more complex than structure. Signal transmission by 7 transmembrane segment receptors. Here you see receptor with its cup-like portion for ligand binding, then 7 segments are shown, and inside you can see G protein. G proteins consist of the 3 subunits. They are designed by Greek letters, alpha, beta, and gamma. And when G protein is inactive, these all 3 subunits are connected to each other and to receptor. When ligand from outside binds to the receptor, G protein becomes activated. And what it means? It means that alpha subunit binds GTP, you see on the scheme, initially it contains guanosine diphosphate. Now it attaches triphosphate GTP and becomes detached from the other subunit. Free alpha subunit activates something in turn. You see, alpha subunit is separated and it binds to the nearby floating somewhere enzyme that also is on the surface of the membrane, so we're submerged to membrane enzyme, which is called generally common effector enzyme. And there are few of such effector enzymes that become activated by G protein. So it goes in the sequence. Then first ligand binds to the receptor. It results in activation of G protein inside. Then G protein activates specific enzyme, effector enzyme. And it's not all the process. So hormone binds, G protein becomes active, which means that alpha subunit separates from the others and binds to the enzyme, thus activating this. And enzyme active starts to produce substances that I mentioned once that as second messengers. Enzyme produces second messengers. The effector enzymes do it. And let's see, effector enzymes are adenylate cyclase. In some sources you may find adenylyl cyclase, which is possible way of naming. But adenylate cyclase, then guanylate cyclase, and phospholipase C. Three main important effector enzymes. First, cyclase name implies that it should form something cyclic. And really, adenylate cyclase produces cyclic adenosine monophosphate, CAMP. You see, ATP molecule is taken as substrate and become converted by the enzyme into cyclic molecule with just one phosphate group, monophosphate. It's achieved through making specific bond, 3' 5' bond. I hope you remember from bioorganic chemistry. So cyclic AMP contains only one phosphate group, but the molecule has cyclic structure. And it's important that this molecule is highly potent in next processes, which we discuss later. Then guanylate cyclase similarly produces cyclic but guanosine monophosphate, cyclic GMP. Also, it takes as a substrate molecule GTP and converts to cyclic GMP. In both cases, two phosphate groups are removed from molecule. But phospholipase C is not a cyclase anymore, you say it's lipase, so it should split some phospholipids. And phospholipase C makes two second messengers at once because it splits one phospholipid molecule into two pieces. And these two pieces are inositol triphosphate, IP3, and diacylglycerol, DAG. And both second messengers, IP3 and DIG, have their next corresponding functions. And what is the function of second messengers? Let's first list second messengers. By now we mentioned only 4 of them, and there is the ancient one, calcium ions. But main are cyclic AMP, adenosine monophosphate, then cyclic GMP, guanosine monophosphate, IP3 inositol triphosphate, and diacylglycerol, and the most ancient calcium ions involved into a number of processes. So 5 classical second messengers. Actually, they are not the only substances that participate in signal transmission. Therefore, some other substances also could be called participants. But classical second messengers are only these 5. And why they are called the second messengers? For what is the first messenger in this case? We consider that hormone itself is a first messenger. It comes to the cell, reaches the receptors, bind to it, and its function is over after binding because it cannot enter the cell. We talk about 7-TMS receptors now only. And then first messenger gives its next task to other substances. And final goal is activation of certain kinases. Similarly to kinases that become activated very quickly and easily in One-TMS receptors, here we also have the task of activation of the protein kinases, certain kinases, different but kinases. And the substance that finally makes it, this final goal, exactly this substance is called the second messenger. Actually, second messengers are not formed themselves. So, for example, cyclic AMP is formed by adenylate cyclase, but adenylate cyclase involved into transmission of signal. It's also helping substance inside the cell, helping structure, but it itself produces second messenger, and only its product, CAMP, which finally activates protein kinase A, is the second messenger, and correspondingly the rest. So cyclic AMP activates protein kinase A, specific kinase that has number of structural of substrate proteins that become converted into active state by protein kinase A. Cyclic GMP activates protein kinase G. It's clear. Then if we skip one line, we go to diacylglycerol, you see it also activates protein kinase C. Perhaps the letter C comes from the protein, from the phospholipase C initially that forms diacylglycerol along with IP3. And the only one exclusion here is inositol triphosphate, IP3. It does not activate protein kinase itself. IP3 molecule formed inside the cell. Its formation occurs on the inner side of the membrane surface, so it's inside. It goes into cytoplasm, for example, in smooth muscle cells, and it binds to the channels for calcium in the reticulum. And so it serves as the substance that opens these channels. And calcium is released from reticulum because inside the smooth muscle cell reticulum, concentration of calcium is very high. So calcium is released from reticulum to the cellular cytoplasm. But actually there is something similar even for IP3, because later we'll study in detail mechanism of contraction of smooth muscle, and you will see that inositol triphosphate releases calcium, but then calcium binds to the specific calmodulin protein, and this complex calcium calmodulin activates certain kinase inside the smooth muscle cell. So finally it comes again to the kinase activation, just here in a longer pathway, not inositol triphosphate itself. So these are functions of the second messengers, and basically this is activation of the protein kinases. Examples of these receptors, 7-TMS and their ligands. Actually, so many ligands operate through these receptors. Adrenergic receptors, or simply we can say adrenoreceptors. Both their subtypes alpha and beta all use 7-TMS receptors. For adrenaline epinephrine and noradrenaline norepinephrine, all are 7-TMS. Then glucagon receptors, ADH receptors, antidiuretic hormone, and many, many others. And it's going to be studied in next lecture. Let's observe again in more detail this whole process. It's very important, very widespread mechanism, pathway, the adenylate cyclase, CAMP pathway of change of cellular function. Here you see plasma membrane and hormone molecule. It binds to the receptor. Here you don't see separately 7 transmembrane segments. We just mean that they are all combined and not counted here. But inside there are clearly 7 transmembrane segments. And on the inner side there is a G protein. For adenylate cyclase CAMP pathway, G protein is specific GS. S stands for stimulating. GS protein. And when hormone binds again, GS protein becomes activated, and it means that its alpha subunit is separated and free subunit moves to nearby located adenylate cyclase. Adenylate cyclase becomes activated and it starts to form cyclic AMP. So it takes ATP molecules, removes 2-phosphate, inorganic phosphate, PPI, it stands for inorganic phosphates 2 group, and ATP becomes converted to cyclic AMP. And it's not only one molecule, not 2, not even 100 molecules, it's many, many molecules which are formed by one adenylate cyclase. And then cyclic AMP activates protein kinase A. It activates by the following way. It binds to inhibitory subunits. Actually, for each one protein kinase A activation, 4 molecules of cyclic AMP should bind to inhibitory subunits, and as they become removed, and inhibition is removed from protein kinase A, and protein kinase A becomes active. And as you may understand, if many molecules of cyclic AMP are formed, many molecules of protein kinase A are activated. So this process takes longer time than One-TMS receptors activation, but it's much more efficient. It's a cascade amplification of the effect. One molecule of hormone is bound, one GS protein is activated, one adenylate cyclase is activated. But cyclic AMP are produced in such amount that the concentration increases 10 or 100 times inside the cell. And therefore, you may imagine how many protein kinase A molecules become active, and they all start to do their function. Activation of inactive enzymes or structural protein change through phosphorylation. And in some cases, we know exactly which proteins become phosphorylated, for example, in heart regulation. Mostly it's the calcium protein, calcium channels protein, and they become open, enabling more calcium to enter the cell, and thus increasing the velocity of depolarization or force of contraction, for example. So, but depending on, in different cells, we have different substrate proteins phosphorylated. Therefore, effects can be multiple, depending on which proteins become changed. In short way, we can sign this pathway as follows. Ligand, the molecule of a hormone, binds to 7 transmembrane segment receptor, and this binding results in activation of GS protein. GS protein in turn activates adenylate cyclase. Then cyclase produces cyclic AMP. Cyclic AMP activates protein kinase A, and protein kinase A phosphorylates certain proteins in the cell, enzymes, structural proteins, and finally we get certain change of the cell functioning. If it's enzyme activation, it results in some changes of biochemistry of the cell. Structural proteins can open channels, can provide many different changes for the cell. Now let's observe similarly the phospholipase C pathway. Phospholipase C, inositol triphosphate mechanism. Here you see only inositol triphosphate is mentioned, but always it's formed together with diacylglycerol. Just we usually omit this. Of course, they both participate. So, receptor protein binds the hormone, and it results in activation of G protein, and now this G goes with index Q. Actually, I have no idea where the Q comes. I didn't find it. But GQ protein is involved into activation of phospholipase C. And phospholipase C becomes active. As you remember, it splits phospholipids according to name. And phospholipids are available, just the plasma membrane consists of them. And among these phospholipids, there is a inositol phosphate. Number 2 indicates that 2 phosphate groups are present. And from this substance, phospholipase C forms 2 second messengers at once. Diacylglycerol activates protein kinase C, not shown in this picture, but IP3 opens calcium channels in endoplasmic reticulum, and calcium is released into cytoplasm. For smooth muscle cell, this activates the contraction of smooth muscle cell. For endocrine cell, it stimulates the process of secretion, exocytosis of produced substances. So this pathway mostly is used for stimulation of smooth muscle cell contraction or secretory processes. Notice that both of them are involved, both pathways involved only in case of 7-TMS receptors. In short, phospholipase C pathway can be signed as follows. Ligand binds to 7-TMS receptors, activates GQ protein, which in turn activates phospholipase C. Phospholipase C produces two second messengers, IP3, inositol triphosphate, and DAG, diacylglycerol. IP3 opens calcium channels, and calcium ions are released into cytoplasm. If it's a smooth muscle cell, for example, calcium binds to calmodulin complex, and then next events occur, which we'll study in future. Diacylglycerol activates protein kinase C, and certain proteins become phosphorylated. So, as a result, we have certain change of cell functioning necessary exactly for this cell. Then, next type of receptors, ligand-gated ion channels. You can see to the left side a closed ion channel, and in the middle you can see an open one, open ligand channel. All the channels composed of protein molecules, of some subunits that are combined as a bunch of these subunits, and they can be very tightly connected to each other so that channel is closed, and they can become wider, more distant from each other so that ions can move. Or sometimes they are exactly like gates formed somewhere in the lumen so that ions cannot move due to these gates closed. And ligand makes the channel to become wider or removes this obstacle gate and makes it gate open so that ions can enter. Here you can see the opening. Left side shows closed channel, too narrow, because binding site is empty. Now binding site is occupied by transmitter molecule and channel becomes open. Why it said here transmitter? Because hormones do not operate through ion channels. Hormones use the other types of receptors, and neurotransmitters usually open channels. Mostly it occurs inside the synapses, in the postsynaptic membranes. These receptors are often ligand-gated channels. Then we can move to the intracellular receptors, but before this I need to remind you that open channel itself does not move anything. If ions have any force for movement, like difference of concentration, so that from higher concentration ions move to the area with lower concentration, or for example, positive ions easily can go inside the cell through open channel because inside the cell is charged negatively. So if there is force that makes the ions to move, so ions will exactly directly move. If channel opens and there is no any difference of concentration, movement will be in both directions, and statistically there will be zero net transportation. Net diffusion will be absent. So channel is example of a passive type of transport. Then we come to the intracellular receptors. Intracellular receptors use lipophilic hormone, bind lipophilic hormones. Here you see combined scheme for both types, cytosolic and nuclear receptors, with little difference. Anyway, first lipophilic hormone should diffuse through the membrane easily because it's dissolved in lipids. Then it can bind, depending on type of receptor, to cytosolic receptor, or directly it goes to nucleus and binds to nuclear receptors. But this difference now is actually lost, because then complex of hormone receptor should bind to DNA. So cytosolic receptor with hormone goes to nucleus and binds DNA. And nuclear receptors are usually already bound to DNA, just they wait for ligand, lipophilic hormone. So, and then when when ligand and hormone complex activates DNA transcription, it's only for specific portion of DNA. So there is specific proteins then can be produced. You see, then mRNA is produced due to activation of transcription of DNA, and then this mRNA is moved to cytoplasm, where with help of ribosomes new proteins are produced. And you may see now that intracellular receptors operate much slower as the receptors such as One-TMS or Seven-TMS, because for both cases of One and Seven-TMS receptors, pathways are different in lengths, but in both cases we use phosphorylation of already existing proteins. For intracellular receptors, proteins should be formed from the zero level, so it takes much longer time to produce new proteins. And thus we can change functioning of the cell. And new protein synthesis requires few hours at least, or even longer, up to days. Signal transmission that involves intracellular receptors can be shown like this. Intracellular receptors that have ligand binding domain and DNA binding domain, they should get the lipid-soluble ligand, which diffuses across the cell membrane and binds to a cytosolic or nuclear intracellular receptors. Next stage is binding of ligand receptor complex to DNA, or if it's a nuclear receptor, actually it's already bound to DNA, just ligand should join. And then it initiates transcription and production of mRNA. Then mRNA is used for the synthesis of new specific proteins. And thus, these proteins can become enzymes, the structural proteins, and other molecules that are able to change the cell functioning. So, the intracellular receptors activation influences the cell through the new proteins synthesis. Studying normal physiology, we are going to learn many examples how chemical signaling pathways produce the following changes. For example, increase the force and rate of heart contraction. Then how blood glucose is regulated through this chemical signaling pathway. How hydrochloric acid production in the stomach can be influenced. Then body metabolism level can be increased or decreased. It can be participation in the learning and memory mechanisms even inside the brain, and many other important processes. Later we'll study this in the course of biochemistry in more details, and we'll see that all this is even more complex. But at first you need to have a clear idea of molecular receptors and have basic understanding of their functioning. So I tried to explain it simply. Now I hope you have an idea, and thank you for attention.